First, before I get started, I'd like to thank my co-authors on this, Phaedra Booty, Mark McKinstry, Peter McKinnon, and Gary Thiede. So many of us are here because we're interested in the conservation of these particular species. And so one of the questions that we ask when we learn that a species is in trouble is, what do we need to know in order to conserve them effectively? And initially, we need to know basic life history questions, as alluded to in the previous talk. What they eat, who eats them, where they can be found in the river, what their habitat requirements are, and so on. And all of these things are influenced and dictated to a degree by how an animal uses space and how they are able to move through it. Therefore, if we know these things, we can answer fundamental questions about their life histories. But none of this happens in a vacuum. Humans have a huge influence on the environment. Down here specifically, we have massive dams like this one at Santa Cacia, which fragments the river. And in the middle of Rio Grande, we have another 2 big diversion dams. And then, of course, the reservoirs at the top and the bottom of the reach. And these aquatic species compete with humans for arguably the most precious resource in the West, which is water. And dams like this allow us to use that. But it causes problems with the habitat fragmentation. That is generally known to be bad, you know, from geomorphological changes to gene flow with the species and so on. In addition to the withdrawals for human use, we also have environmental variables to consider, such as climate change and drought, which can result in massive sections of the river drying up. And when we couple those together, we end up with big restrictions on the movement of aquatic species, whether we're talking about salmon in the Pacific Northwest or silvery minnow down here in the Rio Grande. And so within this context, we need to know how these animals can move within this contemporary fragmented river system, and with the overarching goal of informing future dam passage management decisions and hopefully effective management of the species in general. We all work down here on the Middle Rio Grande, so I'm not going to go too deep into this, but we're specifically working in the Santa Cacia Reach with this beginning part of our study. And our species of interest is, of course, the Rio Grande silvery minnow. They're small, short-lived, and highly fecund. And they used to be one of the most numerous aquatic fish species in the basin, but now they're only found in around 7% of their historic range. And of course, their critical habitat is between the Cochiti and Elephant Butte reservoirs. So we first embarked on a proof of concept pilot study, and we released 1,701 pit-tagged fish of 2 different age classes into the Rio Grande in the spots denoted by the stars. You can see up here is that Santa Cacia Dam. The first release site was immediately below the dam, and then 2 miles and 4 miles further down. And they were released in equal-sized batches. And so once the tagged fish were in the river, we used 3 different styles of pit tag antennas to track their movements and detect them within the river. The first is the floating pit tag antennas, where we hooked up the antennas right here to a raft and attached that to a GPS unit, and we can just float the river and detect them as we go. Then we also have the submersible antennas here, which is a stationary antenna, but you can move them throughout the system as needed, and to put them in places that'll be more effective at collecting your data. And then we also have this wand style, which is kind of like a metal detector that you can use in backwaters to sample areas that are too shallow for your boats. And we came down and did 2 sampling trips that lasted 2 to 3 weeks each, immediately before spring runoff and then immediately afterwards, and used these 2 active styles during those 2-week times, and then left the submersibles out while we were gone. And this approach is really cool because it makes it so that we can see really fine scale movement when we use the active approaches, and spatially as well, spatially and temporally. And then while we're gone, we still have these guys out in the river collecting data on a coarser scale, and so we can still see movement over time while we're not even here. And so with that, what did we see? As I said, 1,701 fish were released, and then this unique detections is how many of those fish that were we able to detect at least one more time. And we detected 979, which was around 58%, which is a pretty spectacular recite rate. And was kind of surprising based on some of the work that we did on the San Juan with these same systems. And then total numbers of detections was over 3,000, which you can translate that directly into movements of fish because we know where they were stocked. And so every single detection translates into a known movement of those fish. And I'll throw in a disclaimer here. All the distances that we report are conservative. We detected them here and here, but what they did in between those 2 points in the river, they could have gone a lot further than that. And I'll start with the obvious. You stock fish in a river, you might expect to see them move downstream. And so I have this split into the 2 age classes, age 1 on the left and age 4 on the right, and the distance moved on the y-axis. And as you can see, there was a difference in the distances that we saw these fish move. The older fish tended to move larger distances downstream. And I broke it down into a little bit finer scale and did it with the fish length on the x-axis and the distance moved on the y, and plotted in the linear regression for that. And you can see there's a trend. As the fish get bigger, they tend to move further distances downstream. And the other thing that you can see here is the maximum distances that we saw in single movements was up around 29 kilometers, which is pretty enormous for a 60 millimeter fish. And so with that in mind, let's look at what they did in an upstream direction. Turns out that the 2 age classes, there was no difference really between the distances that they went upstream. Same plot as before, age 1 fish on the left, age 4 fish on the right, and the distance on the y-axis. They also maxed out at close to 15 kilometers, as opposed to the almost 30 that we saw with the downstream movements. And we show this again with the fish length on the x-axis and the distance moved on the y. And there is, you know, as the regression shows, almost no difference between size classes. And again, you can see how the magnitude of the fish movement is much smaller in an upstream direction. So this made me think, why would that be? So then I looked at the initial movement of the fish immediately after the release. So where did they first go? And how far did they go with that? And so it's broken down by direction moved on the x-axis and then the distance moved on the y. And downstream and upstream are the 2 categories. The upstream category here, as you can see, is quite small with a maximum movement of around 5 kilometers. The downstream is, you know, close to 30. The big difference here is that these fish were limited. They were only released 4 miles below the dam, so that's as far as they could go. And even of those, only a third of them were released that far, so some were released almost at the dam and some 2 miles below that, so that's taken into account there. And then the last thing that I'll show you is the cumulative distance moved by individual fish. So what this shows is how we took the individual movements of every single fish and added them all up together. And so the distance moved on the x-axis is for the total movements of each individual fish. And then the y-axis is the number of fish that moved that far. And the interesting thing, to me at least, was that the median movement was almost 4 kilometers. Which means that even the fish that aren't moving very much, relatively speaking, for this species are still moving large distances, because this was over the span of only, you know, a few months from March to July. And so they are highly mobile fish, you know, as a group, which probably plays into the schooling that was talked about in the previous talk. So with that, I'll get into the preliminary conclusions, because as I said, this was sort of a pilot study. Our approach is highly effective. We had a almost 58% recite rate, which is spectacular. These fish are highly mobile, up to 42 kilometers was the maximum distance moved. And as a group, they all moved quite a lot. And also, these fish have the ability to disperse widely when given the opportunity. So for instance, when there's water in the channel, they can spread out and make use of just about everywhere. They move, as we showed, upstream to the extent possible, and then downstream as well. And so what's next? We're in the middle of our second release event. We just released just over 10,000 tagged fish on the 20th of November. And I just want to thank the Fish and Wildlife Service and BOR guys that tagged those. That was a massive effort and very much appreciated. We're expanding our spatial and temporal scales. We released fish above the Santa Cascia Dam as well, so now they have the ability to go up into the Isleta Reach. And then we are still sampling across all seasons. And then the other thing that our group is working on is some data visualization techniques in order to be able to present this effectively. And with that, I'd like to thank the Bureau of Reclamation for funding, and then everyone else that's listed here. Without you guys, it wouldn't be possible. And if I forgot somebody, I apologize. I would have to defer to Thomas. I don't think there are any plans to tag wild fish just because it's incredibly hard to do that.
Hold on to about 2 weeks because you have daily mortality for about a week, and then there's a lot of shed tags, and that's going into an antibiotic treatment almost immediately. So I mean, you could do it, but you may have to
Can you see schooling with the kind of data you're collecting, since there's GPS tagging and you have the timestamp?
To a certain degree. The problem with that is that there's a phenomenon with pit tags known as tag collision. And so if you have multiple tags within the field created by the antenna at the same time, the one with the strongest signal wins out, and that's the only one you'll detect. And granted that, at least with the boats, you know, you're moving along with the river. And so the time that it would maybe take to see like those effects of schooling and just lots of detections in that one spot are hard to pick up. But with that said, as we float down, we're notified of the tags we detect as we're going. And so there are definitely places where you get, you know, 20, 30, 40 hits, like almost immediately, like within, you know, a minute. So maybe, I don't know if that's just a function of it being preferred habitat or if it's schooling, but.
So how many fish did you find in the second round of sampling versus the first round? Because obviously you floated pretty much immediately, I think, after we released those fish. And then the age thing I hadn't seen before, that was interesting. Were they distributed? I can't remember. Were they distributed evenly at the 3 different sites?
Yeah, the groups that were split, so let's say there was 800 of each group. There was 250 of the age 4 at each place and 250 or so of the age 1 at each place. So they were evenly distributed. Are you referring to the before and after runoff when you mean the 2 sampling events? So the after runoff, we saw much slower detection rate with the boats. We'd still been getting hits on the submersible antennas the whole, like throughout runoff. And part of that may have been that the river was, you know, the flow was about double what we did with the initial release. And so they had the opportunity to spread out into floodplain, and we can't sample in there. It's just, I mean, I can't float a boat in the Bosque.
Have you collected, have you seen any of these older fish yet with the float you just did?
So I haven't had a chance to dig in because we just did a release. So we're actually, my crew's out sampling right now. But I haven't had a chance to dig in and look at like where the sources of the ones that we've seen, but of those 10,000, we're already over, I think we're at like 12 or 15% that we've got detections on again already. So.
And I was just curious, like, because you showed kind of the pool results with the passive and active methods, and you separate those out, and I guess I'm just curious how well the passive ones perform compared to the active. Are they comparable or?
What do you mean by comparable?
I guess just number of detections.
Okay, yeah.
Like, yeah.
So the submersibles, the passive ones, those got about 2/3 of our hits. And then the floating ones had about, around the other third. And then the wand ones that was about 1% of the detections.
What do you see on the fine scale behaviors?
Can you elaborate?
I mean, what do you see?
But what do you mean by fine scale behaviors?
When you're doing the fine scale, did you see the fish move around you or scatter away from you when you're using your wand method?
Yes, you can definitely see that. Theoretically, though, it's not as important for what we're trying to do if they scatter, so long as you still get the detections. And so if you can kind of like herd them up into the backwater and still get it, where they were. And so you can still get the larger movement picture.